Hello student, welcome to EPG Pathshala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about a module called as Evolutionary Aspect in Human Genetics under paper Human Population Genetics. The important points that we are going to discuss in this particular modules are as follows. We will discuss about evolutionary aspect in human genetics, relationship between evolution and genetic diversity. We also try to develop the concept called as evolutionary prediction supported, and ultimately discussing the interpreting evolutionary history related to this particular feature. Let me give you the introductory informations regarding this. The first decades of advances in molecular genetic technology has heralded a new era for all evolutionary studies, but especially the science of human evolution. Data on various kinds of DNA variation in human population have rapidly accumulated. There is an increasing recognition of the importance of this variation for medicine and developmental biology and for understanding the history of our species. Haploid markers from mitochondrial DNA and the Y chromosome have proven invaluable for generating a standard model for evolution of modern human. Conclusions from earlier research on protein polymorphism have been generally supported by more sophisticated DNA analysis. Co-evolution of genes with language and some slowly evolving cultural traits together with the genetic evolution of commensal and parasites that have accompanied modern human in their expansion from Africa to other continent, supports and supplements the standard model of genetic evolution. The advances in our understanding of evolutionary history of human attest to the advantage of multidisciplinary research. So, we can say that reconstructing human evolution requires both historical and statistical research, although conclusions are not experimentally verifiable because the process cannot be repeated. Various disciplines such as physical and social anthropology, archaeology, demography, and linguistics provide complementary approach to researching question of human evolution. The existence of molecular genetic variation among human population was first demonstrated by Hirschfeld, and Hirschfeld in a classic study published in 1919 of the first human gene to be described, ABO, which determined ABO blood group. The subsequent identification of blood group protein markers such as MNS and RH expanded the repertoire of polymorphic markers that could be analyzed using antibodies. Fisher showed that evolution could be reconstructed by analyzing the multi-locus genotype on a chromosome observed in population and their inheritance within the families. The term haplotype for the multi-locus combination of allele on a chromosome was introduced by Cipollini et al. During early research on the major histocompatibility complex. Immunological method remained the only satisfactory technique for detecting genetic variation until Pauling et al. Introduced electrophoresis to separate different mutant of hemoglobin, a technique that was rapidly adopted to analyze variation in other blood protein. It was soon obvious that genetic variation was not rare, but on the contrary that almost every protein had genetic variant. These variants became useful markers for population studies. The first book of allele frequency in population published in 1954 was limited almost completely to serological variation, and the books listing genetic variation increased rapidly in size and number. In 1980, a method of studying variation in DNA identified mutant of restriction sites by using a radioisotope and generated several new markers. But it was only with the development of PCR in 1986 that the study of more general DNA variation became possible. The development of automated DNA sequencing in the early 1990s paved the way for the application of systematic study of genome variation to human evolutionary biology. Data from protein markers are still more abundant than are data from DNA, although this situation is rapidly changing. Nonetheless, molecular genetic markers have provided previously unavailable resolution into question of human evolution, migration, and the historical relationship of separated human population. In this review, we will discuss the evolutionary and historical forces that have shaped genomic variation and how its interpretation has led to a deeper understanding of the evolution of our species. Evolution and genetic diversity. In 1800s, the lumper potatoes grown for Irish, that is in 1800, a potatoes clone called lumper was grown for Irish. Similarly, in 1840s, potato blights hits, and during that period, all potatoes were susceptible to this potatoes blight. This explains the evolution relationship between genetic diversity and evolution, as in there is a cases of 1 in every 8 Irish peoples were died during that particular period. So because of the genetic diversity, even though there is a clone was developed, a clone of potato was developed, but when but later on it was found that this clone has a, instead of having a positive effect, it has somewhat degraded effect, and the result was so astonishing that almost out of 8, 1 people died because of this particular feature, because of this diversity. So then genetic diversity is key to surviving diseases. Evolutionary prediction supported. There is a table which indicates the amino acid reveal evolution. Cytochrome C evolution. We will see the number of amino acid differences from human. Chimpanzee, there is no differences. If we see the rhesus monkey, there is only 1 amino acid differences. Rabbit 9. In the case of cow, it is 10. Then pigeon 12. Bullfrog 20. Fruit fly, see the difference between mammalian and fruit fly, 24. Wheat germ 37. Yeast 42. So the difference between in the case of number of amino acid from human being is 42 in the case of yeast cell, and 20 in the case of bullfrog, 9 in the case of rabbit, rhesus monkey only 1, but there is no differences in comparison to chimpanzee. So now let's discuss about its perspective. Even though we have seen that there is a close similarity between human and chimpanzee, but human and chimps are quite different. Not very much, but they are different morphologically as well as physically, or we can call phenotypically. Even though genetically they have a close similarities. So human chimps are different. Example, we are much smarter in comparison to chimpanzee. So there is no reason to be ashamed of a common ancestry with chimps. Remember, certain apes are not our ancestor. They are more like our cousin. So there is two. One may be direct, they are directly connecting, called as our common ancestor. Then some other group of chimpanzees, they are like our close cousins. They evolve along with us. The rapid change, especially in the case of brain size, can occur with small changes in genome, even though the process is highly complicated. So the perspective clearly indicate that there is a difference between human and chimpanzee, but there is a lot of similarities too. Because of that, we can say that they are known to have, that we too have a common ancestry, and some of the advanced variety, some of the advanced species of chimpanzees, they are known to be our close cousin. Testing the evolutionary hypothesis of common ancestry. When we test the hypothesis related to common ancestry, the chromosome number in the great ape, we can see the comparison between human, chimpanzee, gorilla, orangutan, what we call it homo, pan, gorilla and pogo. See, in the case of human being, there is 46 chromosomes, and chimpanzee, gorilla and orangutan 48 chromosomes. Because of their very similarities, we will see that they are known to be our are they are our ancestors, or we too have a common ancestry. So testable prediction is as such, if these organisms share common ancestry, the human genome must obtain a fused chromosome. So there is a great possibility that they may have a common ancestry, or they are known to be our lineage, or directly they are related to our to us in the evolutionary pedigree and the evolutionary phylogenetic feature. It shows the number of DNA nucleotide based differences in the cytochrome oxidase gene. If we see the differences between human, pig, duck, snake, tuna and moth and even the yeast, the number of DNA nucleotide based differences is only 13 in the case of pig, 17 in the case of duck, 20 in the case of snake. In the case of tuna, it is 31, 36 in the case of moth, whereas it is 66 in the case of youth. So conservation at the molecular level, why else should the different organisms possess related genes, is explained in this particular diagram. It shows why does the degree of relationship of genes match their degree of relationship established by other matter. So the base total is 312, and if we see the relationship, there is a great similarity as well as differences, and the distance clearly indicate the number of DNA nucleotide based differences in the cytochrome oxidase gene. Interpreting evolutionary history. Here in this slide, it shows the summary tree of world population. Phylogenetic tree based on polymorphism of 120 protein genes in 1915 population ground by continental sub-areas and first gene genetic distance. Root placed assuming a constant rate of evolution. So the history of population differentiations using genetic data was initially inferred from phylogenetic tree and from multivariate statistical methods such as a principal component of which multidimensional scaling is a derivation that use allele frequencies. Population tree are especially useful for reconstructing history if population differences can be assumed to result from fission that occur randomly in time with a constant rate of neutral evolution in each population between fission. This is likely to be roughly true for data on several autosomal genes from large populations that are geographically and genetically distant, as illustrated in figure 1, which shows 9 such groups from around the world. Completely different types of DNA variations provide the same basic conclusion regarding the relationship between these populations. Violation of the above assumption, such as the presence of migrations or selection, affect the interpretation of population tree. However, when migration, so in this particular slide, relationship between genetic and geographic distance, genetic distance of population pair, it measures by first as a function of geographic distance between members of the pair. Only samples from the indigenous peoples were included. Continents where primitive economics predominant, hunting gathering or tropical gardening shows highest symptosis, highest asymptotes. Asia and the world do not asymptote within the range zone. So however, when migration between geographic neighbor is frequent, principal component displayed in a two-dimension reflect the geographical distribution of population. Under the simple evolutionary model described above, tree and principal component gives similar result. For populations that are geographically close, genetic and geographic distance are often highly correlated, with a asymptote for the genetic distance at about 1000 to 2600 miles on average, but higher for Asia and the world, which are not at equilibrium. Recent statistical development in detecting clustering among population based on highly polymorphic autosomal markers have been valuable for analyzing very large population genetic data sets. It is important that this completely different approach produces the same primary continental clusters as the earlier method. In its application to datasets with numerous polymorphic loci, however, it does seems to be more sensitive in detecting and assessing individual ancestry. Now let's try to tracking migrations of our species using DNA. A recent synthesis of Y chromosome, phylogenography, paleoanthropological and paleoclimatological evidence suggest a possible hypothesis for the evolution of human diversity. Around 100 kya or shortly after a small population of about 1000 individuals, that is a tribe, most probably from East Africa, expanded throughout much of Africa. Then between 60 and 40 kya, there was a second expansion, most probably from a descendant population into Asia and from there to other continents. This may be referred to as the standard model of modern human evolution. It is also called out of Africa in recognition of earlier expansion of Homo erectus from Africa into Eurasia of about around 1.7 million years ago, and assume that anatomically modern human replaced earlier poorly known species of Homo that descended from the first migrant of Homo erectus. Genetic data provide some indication that the spread of human into Asia occurred through two routes. The first was a southern route, perhaps along the coast to South and Southeast Asia, from where it bifurcates north and south. In the south, this modern human reached oceanic between 60 and 40 kya, where the northern expansion later reached China, Japan and eventually America. This might represent the second migration to America, associated with Nadine language postulated by Greenberg. The second was a central route through the Middle East, Arabia or Persian to Central Asia, from where migration occurred in all directions, reaching Europe, east and northeast of Asia about 40 kya, after which the first and the principal migration to America suggested by Greenberg occurred not later than 50 kya. It is still unresolved whether the divergence between these two expansion routes occurred in Africa or after entry into West Asia, and if the latter where it happened. Most literature accept without discussion that the entry to Europe and Central Asia was through the Levant. It is not at all certain that this was the only or the earliest route. These two initially divergent routes converge later, especially in the extreme East and America. An alternative to the out of Africa hypothesis originated by Weddenrick and expanded and called multi-regional by Wolpoff maintain that all human population living today originated in their various continents and evolved in parallel into modern human. The main basis of this hypothesis is the claim that most ancient fossil, especially those from Europe and Asia, but not Oceania and America, where the human fossil founds are all very recent of modern human type. They show a continuous morphological transitions to modern human. An extreme example of parallel evolution that include the doubling of brain volume is invoked to explain this scenario. In a later version of a multi-regional model, parallelism is claimed to be the result of a substantial intermigration. It shows the high-resolution molecular phylogeny to study human history. A phylogeny of human mitochondrial DNA haplogroups and their continental affiliations composed from resequencing of 277 individuals. The length of the branches correspond approximately to the number of mutations. So, the identification in recent years of a large number of SNPs on the NRY and mitochondrial DNA has afforded higher resolution of population history through the reconstruction of a phylogenetic relationship of extant Y chromosome and mitochondrial DNA. Using the nomenclature developed by the Y chromosome consortium 114 and the first two haplogroups are almost completely African and even today represent mostly hunter-gatherer on their descendants who have never reached high population density or undergone high rates of increase. So, it's like in the case of it shows slow growth is indicated by the accumulation of many mutations within a branch, as in most descendant of haplogroup A and B, and in those of earliest branches of haplogroup C, D, E, and F. By contrast, when there are many branches called a starburst after a specific mutation or group of mutation, we can infer rapid growth. The major expansions are those haplogroup F, 7 branches, after an initial lag in population growth. And even more remarkable is the later expansion of haplogroup K having 9 branches. These began in the last 40 KYA and led to a major settlement of all continents from Africa first to Asia and from Asia to the other 3 continents. The tree of mitochondrial DNA as shown in figure is more bushy, but there are more haplogroups because of the higher mutation rate. The general structure of the male and female genealogies in figure shows are the same. The earliest branch all remain in Africa. In both trees, they clearly refer to the slowly growing hunter-gatherer. In both trees, the major growth in Africa is due to a late branch taking place in the second part of the last 100,000 years and clearly connected with the expansion to Asia. The MN branch of mitochondrial DNA phylogeny indicate the separation of the expansion from Africa to Asia into a southern and northern branches. In the NRY genealogy, the southern branch is on average earlier than the northern and include mostly haplogroup C, D, H, M, and L. Of this, H and L remain in India and part of C went to Oceania. The rest to Mongolia, Siberia, and eventually to northwestern America. D went as far as Southeast Asia and Japan. The Northern Asian expansion remained mostly in East Asia. Haplogroup O its branch and as a major propagule to northeastern Europe among the Uralic speaker. Haplogroup I from North Asia generate what is probably the first major Paleolithic expansion to Central Europe. G and J are found today in the Middle East and from there expanded to Europe, mostly in the south and probably with Neolithic farmer. R is found in Europe, India, Pakistan and America, but an early branch seems to have returned to the central part of Sahel in North Africa. Haplogroup Q generate most Armenids, except for Nardini speakers and Eskimos. Haplogroup I is also found in North and Central Europe, where it probably originated around 20,000 KYA. A few indigenous individuals in America and Australia probably inherited European Y chromosome. It shows the high resolution history using haploid markers as described earlier also. Here in this case, language families of the world, 12 families of the Greenberg classification. The Eurasiatic superfamily include 6 families, most of which are recognized by most linguists and is an isolate Gilead listed in the central column. The oldest family is the Khoisan that include Bushman and Hottentots, many of which also belong genetically to the oldest haplogroup of both mitochondrial DNA and NRY. Australian and Indo-Pacific are also old families. Other African languages are Niger Kordofanian, mostly West Africa, Nilo-Saharan, and Afro-Asiatic that include Semitic languages like Arab and Hebrew. American language belongs to 3 families. Americans were the first to migrate from Asia, according to some as late as 15 KYA, and American shows affinities with Euroasiatics, one of the other 2 Euroasiatics, and include Sino-Tibetan spoken in almost all of China as well as some isolated, probably relic language, Basque, a few Caucasian language, and Burusaki spoken in northern Pakistan, that all survived the later spread of Euro-Asiatic language. The third American family is Eskimo Aleut, the last to spread to America from northeast of Siberia. The Austric family is very large and is spoken in Southeast Asia, Indonesia, all of Polynesia to the east and Madagascar to the west. So, it has been classified, what we call it, a general classification based on 12 language families has been suggested by Greenberg. For geneticists like us, it seems natural to think that modern languages derived mostly or completely from a single language spoken in East Africa around 1000 KYA, given the today's gene also derived from that population. This does not mean that this was the only language in existence at the time. In parallel with the genetic TMRCAs, it was the only language then existing that survived and evolved with rapid differentiation and transformation. Evidence supporting the existence of a common single language include the shared lexicon, sound, and the grammar of present-day language. The expansion of modern human may have been stimulated by the development of new, more sophisticated culture of stone tool called organizations, which developed at the time of the expansion. It is also very likely that navigation became available, or else the passage from Southeast Asia to Oceanic would have been impossible. and may even have been used earlier, such as in coastal South Asia or later along the Pacific American coast. Innovation that increased food availability may have then allowed group to remain in the same area and to increase in size. This apparently happened in many parts of the world on a massive scale starting 10 to 13 KYA with the adaptation of agriculture and pastoralism. From the beginning of food production to the present, there must have been a thousand-fold population increase. Demographic growth in the well-identified specific area of origin of agriculture must have stimulated a continuous peripheral population expansion wherever the new technologies were successful. Demic expansion is the name given to the phenomena that is farming spread by farmers themselves, as contrasted with cultural diffusion, that is, the spread of farming technique without movement of people. Innovation favoring demographic growth would be expected to determine both demic and cultural diffusion. Recent research suggests a roughly equal importance to demic and cultural diffusion of agriculture from the Near East into Europe in the Neolithic period. Hence, humans carry many different kinds of commensal organism or feature which is cultural in nature and which has its direct role on the transmission, transformations and development of feature which explain the high resolution history using different kinds of markers, DNA and other features. Now let's discuss about the important point in the form of summary. As we have already discussed that population genetics provides a model for investigating the balance of evolutionary force acting on genetic diversity. Genetic evidences strongly support evolution. There are studies that use this model have found that the evolution of contemporary human genetic diversity has occurred over the past several 100,000 years or longer. Our species is geographically widespread but shows low level of differences among population groups, suggesting persistent level of gene flow as well as dispersal. It is difficult to classify human into group by their DNA profile and impossible to successfully apply a biological concept of race to diversity within living human population. Thank you.